Hello, everybody. Welcome to Cannabis Grand Rounds. Okay, we'll just wait a couple more minutes. And as we wait, I just want to make a couple of announcements. The first is just a reminder that we will not be having a faculty meeting next week. We will be having our next faculty meeting on September 22nd from 12 to 1. More people will be back then for the fall quarter. So we're looking forward to seeing you. We're still going to do it over Zoom for right now. We'll let you know when we start doing the faculty meetings as hybrid. Hopefully that will happen soon. The next announcement is we are having an event on October 7th, Friday, October 7th, from 9 a.m. till 2 p.m. And this event will focus on the grants that have been awarded from the Department of Cannabis Control. So we're really excited to be able to feature the studies that are underway, looking at the impact of Proposition 64 on health outcomes. And those are the main announcements. We're so excited to have Dr. Verrico here today. And before I introduce him, for people who have questions throughout Dr. Verrico's talk, please feel free to click on the question and answer button underneath the tiles. So it's really wonderful to have Dr. Verrico here. Many of us know him from the drug-dependent research world. He did his undergraduate training in neuroscience from the University of Pittsburgh, and then followed it with a PhD in pharmacology from Yale, where he concentrated on cannabinoid-induced cognitive impairments. He then followed this with 10 years of postdoctoral training at Harvard, Carnegie Mellon, and the University of Pittsburgh that led him through researching cannabis in the adolescent brain, MDMA impairment in primates, and finally researching antipsychotic drugs and control of dopaminergic neurons. Right now, Dr. Verrico is an associate professor in the departments of psychiatry and pharmacology at Baylor, and he's also a principal investigator at the Michael DeBakey VA Medical Center, and it's there that he conducts human laboratory studies and outpatient clinical trials. Chris is a principal investigator on awards from the NIH, the Department of Defense, and the McNair Foundation. And 2 awards he was recently given just last month. One, R01, is testing the effects of extended release exenatide and nicotine replacement therapy on smoking cessation and post-cessation weight gain. And very much applicable to this talk, he was just awarded a grant to understand the efficacy of cannabinoids for decreasing the intensity of endometriosis and back pain. Dr. Verrico has received numerous honors, including one recently from Baylor titled the Ally of Women of Excellence. He teaches, facilitates a journal club for psychiatric residents, and mentors medical students and residents and other trainees, and has been very successful, and they've been productive with over 25 publications published by these trainees and 26 presentations at international conferences. So today we're going to hear a little bit about Dr. Verrico's 25 years of research in the field of cannabinoids that has recently become increasingly recognized. His work has taken him through academia and industry. He's given tens of presentations on cannabinoids internationally and locally, and has published 16 peer-reviewed publications on cannabinoids in many high-impact journals, including American Journal of Psychiatry and Neuropsychopharmacology. We're so excited to have Dr. Verrico here today to talk on the topic of 25 years of cannabinoid pharmacology, experiments across species with an eye on medication development. And with that, Dr. Verrico, I'll turn it to you so you can go ahead and share your slides.
Great. Well, thank you for the intro, and happy to be here. So if I can you all see that?
Perfect, yes.
Okay, great. So yeah, I'm gonna take you on a bit of a journey of my life. And I'm actually gonna start pretty early. And so if you grew up in the 80s, you may remember actually watching music videos on MTV, or passing notes in class rather than texting, playing Pong on Atari, or listening to mixtapes on a Walkman. what made a huge impression on me and a lasting impression was when on one of my favorite TV shows at the time, the sitcom Different Strokes, Nancy Reagan came on and she shared her "Just Say No" message. And so why that made a big impact on me was because that and other related messages, such as "This is your preying on drugs," about the dangers of using illicit drugs, just seemed a little simplistic. It seemed to demonize everything from cannabis use to angel dust. And this just didn't really match what I was observing firsthand when I was in high school. And so rather than providing accurate information so that kids could make an informed decision, these messages really did nothing more than spread fear and ignorance. In fact, what we know now is that the optics actually proved quite costly. And so at the time in the mid 80s, about 2 to 6%, 4% of Americans saw drug abuse as a #1 problem. But that jumped in the next 4 years to 64%. And this led to President Reagan signing the Anti-Drug Abuse Act in 1986. And this allotted $1.7 billion to continue the fight on drugs, or the war on drugs. It also established mandatory minimum sentences for specific drug offenses, and included the what was known as the 100-to-1 I can't say that word, ratio. And so, for example, a minimum penalty of 5 years was given if you had 5 grams of crack cocaine, or if you had 500 grams of powder cocaine. That's what I was trying to get at. And this proved to be quite costly, as the number of people incarcerated for nonviolent drug offenses skyrocketed from 50,000 in the 80s and to 1997, where it's about 400,000, so 8-fold increase. Not only was it costly, it turns out that having authorities describe the dangers of drugs and alcohol abuse and encourage students to flatly reject peer pressure doesn't have a substantial impact on long-term behaviors or perceptions. And so a 1990 study on the impact of Project DARE and 1,002 sixth graders who received DARE and were then reevaluated at the age of 20 found few differences between them and other sixth graders who received a standard drug education curriculum in terms of actual drug use, drug attitudes, or self-esteem. And in no case did the DARE group have a more successful outcome than the comparison group. But for me as a teen, I didn't have to wait 10 years to find out that this type of messaging was ineffective. It simply didn't match reality. That this implied danger of drugs like cannabis wasn't matching what I was observing firsthand among my peers. And so what I wanted to know then was, what do they know and what don't they know? Because it was never clear to me what the foundation was for making these sorts of claims. And so the answer for me was education. And this is what, in fact, motivated me to study neuroscience at Pittsburgh. And, I was fortunate that at the time, and still, there are some leaders in the field there, such as Tony Grace, Susan Cisak, and others. And they taught me about how, drugs affected the brain. It's from there, I then went on to study pharmacology at Yale, which is when I began looking at the effects of cannabinoids. And this was under the supervision of mentors, including Bob Roth, Tony George, who presented at this same meeting not too long ago, David Yench, and Rich De La Garza. And so my focus on cannabinoids was driven by the fact that so many people used it, including groups of friends of mine, while our knowledge as scientists wasn't clear how cannabinoids were even producing their effects, especially in the 90s. And so that's just a little brief background as to why I even started looking at cannabinoids, why they were of interest to me. And so today I will start by telling you about my thesis project at Yale, in which we looked at the effects of THC in a rodent model. Then I will tell you about the work I did in Pittsburgh in adolescent rhesus monkeys. I will then, well, maybe not in this order, but then I will tell you about a cannabidiol study we did in canine osteoarthritis, and go on to tell you about a human medication study that built upon the rhesus monkey work and in humans. And then if there's time, I will briefly tell you about a upcoming study we have to look at cannabidiol and THC as treatments for chronic lower back pain and endometriosis pain. Okay, so going back to 1996, and I was a young graduate student, and at the time, I just want to give you a little perspective of what we knew at the time. We didn't know much. It wasn't until 1984 that a THC binding site in the brain was identified. And then it wasn't until the 1990 that the CB1 receptor was actually cloned and sequenced. Then came the identification of the endogenous cannabinoids, including anandamide in 1992 and 2-arachidonyl glycerol in 1995. Subsequently, the enzymes required for both the degradation as well as the biosynthesis of endocannabinoids were beginning to be elucidated. With an understanding of the endocannabinoid system, a speculative role of cannabinoid dysfunctional signaling in schizophrenia began to emerge based on observations that a high rate of marijuana consumption had been associated with increased chances of having some psychiatric episode throughout a lifetime. And additional support came from studies that revealed high acute doses of THC produced both visual and auditory hallucinations, delusional ideas, thought disorders, symptoms of hypomania in normal volunteers. Also, the fact that marijuana appears to be somewhat unique in its ability to produce these negative or deficit state symptoms, such as social withdrawal, and that the cognitive impairments induced by THC were like those exhibited by patients suffering from schizophrenia. And so together, these data suggest that marijuana use may induce schizophrenia, as the three primary symptoms, the positive, the negative, and the cognitive impairments, are all simulated by cannabis. And so while animal studies had consistently shown up until then that acute administration of a CB1 receptor agonist impaired hippocampal-dependent memory processes, the effects of cannabinoids on prefrontal cortical mediated cognition were largely overlooked. However, there were a few human studies that had been done, including one showing that the lower regional brain flow values in prefrontal cortex and other frontal areas after long-term cannabis use, and that chronic marijuana smokers showed diminished activation in the dorsolateral prefrontal cortex, and that this persisted for a month, 28-day period after they stopped using. And that repeated THC exposure to rats induced persistent deficits in a prefrontal cortical dependent spatial working memory task, but not on non-frontal mnemonic functions such as reference memory. And so taken together, these clinical reports, coupled with the behavioral studies in rats, were consistent with the theory that THC causes persistent dysfunction of prefrontal cortical systems. However, at the time, relatively little was known about the possible neurochemical basis of these effects. And this is what takes us to my rodent work. So since repeated THC administration is causing these enduring behavioral and cognitive deficits in animals and humans, and because the prefrontal cortex is particularly sensitive to perturbations of mesocortical dopamine, we focus these investigations on that innervation of the rat forebrain. And so specifically, we sought to determine if the repeated administration of THC affected the dopaminergic innervation of the rat prefrontal cortex. And so in this study, rats received either 5 or 10 milligrams per kilogram of THC twice daily for 14 days, while the control subjects received a saline with a drop of 80, which is what we use to reconstitute the THC. And then after a 7-day washout period, the animals were sacrificed, dissected out their brains, and looked at dopamine turnover in the prefrontal cortex, as well as the other targets of the midbrain dopamine neurons, including the nucleus accumbens and the dorsal striatum. And so what we found here, what you're looking at on the x-axis is just number of rats in each group, and on the y-axis is just the dopamine to dopac to dopamine ratio relative to controls. And so what you see in the white is the vehicle-treated animals. In the middle bar, wait, there is like a laser here, huh? Right. And so here, we're looking at 5 milligrams THC, and then the far right is the 10 milligram. And you can see that there's a significant reduction in dopamine turnover following 10 milligram dosing. However, when we looked at the nucleus accumbens, we saw nothing in either THC group, and this was also consistent with what we found in the striatum. So the only other thing I'll add here is that, it did not appear as though that the THC was toxic to the dopamine neurons, since the levels of dopamine concentration of the prefrontal cortex were not effective. So it suggests it is affecting the actual release of dopamine. So from here, we knew we needed at least 10 milligrams per kilogram twice daily to cause this sort of neurochemical change. Next, we wanted to know, can we use a shorter duration of treatment with THC, or can we use a synthetic CB1 receptor agonist? And in this particular case, we use WIN 55212 to see if it produced similar effects. And we also wanted to see if the effects persisted beyond the 7-day withdrawal. And so we administered THC or WIN, 10 milligrams per kilogram of each. And that was done for either 7 days or 14 days. And then after either a 7-day or 14-day withdrawal period, we again evaluated dopamine turnover in the prefrontal cortex and the other brain regions. And so you're looking at the same sort of graph here, where it's just animals on the x-axis, and on the y-axis, we're looking at the dopamine to dopac to dopamine ratio. And I'm only going to show you the results for the prefrontal cortex because, like the prior study, we did not see any effects in the nucleus accumbens or in the striatum, dorsal striatum. So here you can see, though, that we found that the 7 days treatment with either THC or WIN reduced the dopamine turnover in the prefrontal cortex, but THC for 7 days did not reach significance, whereas WIN did. And I'm sure you all know, I didn't at the time, but now we know that, well, at least now I know that THC is a partial agonist of the CB1 receptor, whereas WIN is a full agonist. And so that might explain why it was more, it didn't require quite as long exposure. Here we're just looking at, basically this is a replication of the study I just showed you when you look at the prefrontal cortex here, but we see that WIN reduced it as well. And finally, looking at 14 days post cessation, we see that the effects persisted for at least 14 days of withdrawal, although there was clearly some recovery as well. Okay, so after this, the next thing we wanted to do then was use this exact same dosing regimen, 10 milligrams per kilogram twice daily for 14 days to induce this persistent dopaminergic deficit in the prefrontal cortex. And because dopamine is known to affect attentional processes and other executive functions associated with the prefrontal cortex, we wanted to examine the cognitive consequences of this THC regimen. And so specifically, we sought to determine if it impaired the acquisition as well as the performance of a self-paced serial reaction time task. We also wanted to see if we could reverse this effect. And so we had an acute challenge with an amphetamine dose. And amphetamine is known to increase catecholamines in the prefrontal cortex, and that's why we chose that. So for these studies, actually, before we administered THC, I surgically implanted a polyethylene catheter into their intraperitoneal cavity, and then tunneled it subcutaneously up at the neck. And this allowed for us to administer the drug without inducing any sort of stress, because stress was known to affect performance on this task. And so after a 3-day recovery period, then we began administering the THC for the 14-day period. So then during the first 7-day washout period is when we trained the animals on the task. And you can see here that there's in front of the rat, there's a panel that had 5 apertures, but 2 of them were blocked off. And so there was only 3 that they had access to. And so in this task, then rats are required to take a pellet from the magazine to start the task. And then the light above the magazine was extinguished. 3 seconds later, the center aperture light was illuminated, and the rat was required to complete the nose poke for a variable duration. And then either the far right or the far left aperture light was illuminated. Here it's the far right. And there was a target stimulus there, and that varied across the 4 days of acquisition, such that it was 30 seconds, then 5 seconds, 2.5 seconds, 1 second on days 1, 2, 3, and 4. After the target was delivered, a correct nose poke during the illumination period resulted in all the aperture lights to extinguish and a pellet to be delivered to the magazine. So rats were then tested on this paradigm where we used a random stimulus duration of 4, 1, 2, 1, or a half a second. We then waited another 7 days. We tested the animals again, both before and after the acute amphetamine challenge. And so the dependent measures were things like accuracy, obviously omission errors, commission errors, etc. But I'm only going to show you the accuracy. So during the acquisition phase, you can see here, there was a main effect of THC exposure on accuracy. And this did not interact with the stimulus duration. And as might be expected if it's a purely attentional deficit. And while I said I won't show you them, I'm going to let you know that all the other measures we looked at were not affected. And then during the test phase, here you're looking at the control animals after a saline challenge, and then the control animals after receiving the amphetamine challenge. It didn't affect their performance at all. But we see that the THC administration followed by a saline challenge, the attentional deficits persisted, but we were able to reverse that with this low dose of amphetamine. I believe it was 0.5 milligrams per kilogram. So just to quickly summarize this then, what we saw was that cortical dopamine function was reduced after repeated THC administration. Repeated the same dosing regimen that induced those dopamine deficits also impaired cognitive performance on an attentional task. And this all persisted for at least 14 days after THC administration had stopped. So there are clear limitations to this work, as most work. And, one of the key limitations is the dosing schedules that are really not comparable to what humans use. 10 milligrams per kilogram is a considerable dose, let alone the difference between species. So before I get into the monkey stuff, from there, I went to work with monkeys at Harvard's New England Primate Research Center. And here I studied the effects of MDMA in monkeys and looked at cognitive performance. And I worked with Bertha Madras and Greg Miller. It was after that that I went to Carnegie Mellon. And there I was learning how to record from single neurons in the awake behaving monkey while they were performing a cognitive task, working memory task. My goal was to actually use the electrophysiological approach in monkeys to determine how THC was affecting these mechanisms of information processing. I was there for 1 year and I had finished my first monkey and, recorded from probably, it was in the 90s. I don't remember exactly how many neurons I recorded from, but it was good data. And I was just starting on my second monkey when Sanofi Aventis relentlessly recruited me. And if you don't know, Sanofi Aventis had the CB1 receptor antagonist, which I always knew as SR141716A, but was called rimonabant. They had it approved in 56 countries as a treatment for obesity. And so they wanted somebody to come in, set up a monkey model so that they could continue to look at these types of drugs to see if there was any potential for them to enhance cognitive performance. And so that was my task to, get a monkey model where we could actually see improved performance. However, a month after I got there is when the reports of serious psychiatric side effects started to come in. And that included things like suicidality. And that concluded Sanofi Aventis's foray into the cannabinoid development program. And they discontinued rimonabant and they discontinued their interest in pursuing cognitive enhancers. And they gave me the option to study self-administration of different compounds they were developing, which was not of interest to me. So I actually resigned and did not have a job for several months. But I was very fortunate. Because I was born and grew up in Pittsburgh, I went back to Pittsburgh. And while I was landscaping during the summer, I went back to CMU and talked to Carl, my advisor at the time, and he put me in touch with Dave Lewis at Pittsburgh. And Dave has always had an interest in schizophrenia. And because of the connection with cannabis, he was working on an R01 and it had received the score. He was putting in a resubmission. And so I helped with that. And then I came on board. And so at the time when I started these studies, there was this increasing reports about how adolescent cannabis use was substantially increasing the risk of schizophrenia. And that's what you can see here in this table. Is that, chronic cannabis abuse, the effect size as far as a risk factor for schizophrenia, it's as great as there anything else besides being besides genetics. So we conducted these studies in adolescent monkeys. Before I get to that, I'm going to tell you what I mean by adolescence. And so typically, it's associated with the teen years, maybe 13 to 19 years old. But I'm going to tell you why I think it's actually a little bit longer. And that's not just my thought. I think a lot of people would agree. That's in part because the executive functions that are mediated by the prefrontal cortex in adults continue to improve during adolescence, right? And so things like cognitive flexibility, which is the ability to think about something in more than one way, and requires set shifting type abilities. And so the interdimensional, extra-dimensional shift task is one example of that. Continues to develop, as does inhibitory control. And so this is needed for self-monitoring, ignoring distractions, things like the Stroop task, where you have to inhibit saying the color of an object and instead focus on the letter itself. I didn't explain that well, but you'll see in a second. And then there's working memory, of course, which the typical task, well, one of the traditional tasks for measuring working memory was a Wisconsin card sorting task. And so cognitive flexibility appears to actually mature to adult levels of performance a little earlier in adolescence, by around age 15. Inhibitory control is thought to stabilize a little bit later, maybe like age 16 or 17. And I would just highlight that even though, children can inhibit a response, their efficiency in applying that knowledge consistently is what improves over adolescence. And then working memory begins to peak in late adolescence, with spatial working memory peaking last during young adulthood. And so that's why I have placed adolescence in humans going out to about 22 years. And so this stepwise improvement in the executive functions is thought to reflect maturational events in the frontal areas. And so here, this is an example of a self-ordered spatial search task. And so what happens is there's like a grid such as this, and then a number of different colored boxes come up, and they represent where a participant needs to search for the presence of tokens. And so only one token is hidden under each of the indicated locations, and they have to remember where each of them is and not revisit any ones they already have. And so it requires generating like a strategy, updating information, and monitoring performance. And so here it just goes through a quick example of different locations being identified, and then having to accurately recall, and then you would receive a reward or a token. This type of working memory, spatial working memory, is mediated through the dorsal stream. And so from your visual cortex, it's going through your parietal cortex before landing in the dorsolateral prefrontal cortex. And what we see is that accuracy on this type of task improves until adulthood. And so you see an age-related increase in performance. You also see that activity within the dorsolateral prefrontal cortex increases as well. And so there's this positive correlation with working memory and working memory related activity. So in contrast to this, might be something like an object familiar task, a delayed match to sample, non-match to sample. And by familiar, I mean that the same objects are used trial to trial. And so as an example, showed an image of a razor, and then after a delay, you'd be shown that image as well as a new one, and you'd have to remember which one you saw. And then this next trial uses the exact same two objects. And so you have to remember which one you've seen most recently. And this yields, this requires the cognitive flexibility that I discussed a second ago, and similar to inhibiting the, well, let me just move on. So this actually goes to the ventral stream. And so in through your visual cortex, it stops at the hippocampus and medial temporal lobe. But because the objects are familiar and you have to inhibit responding on some trials to what you've seen on the last trial, it requires additional cognitive effort. And so the information goes on to the ventrolateral prefrontal cortex. And what you see on these type of tasks is that while children perform below the level of teens as well as adults, the plateau occurs during the teen years. And we also see a very similar pattern with the activity of neurons in the ventrolateral prefrontal cortex. what's the difference between that and an object unique task is that the unique task is always using different stimuli on each trial. And so this is an example, you're shown one image, and then you have to remember that one image when you're shown two more. And then a new image appears, and you have to remember that one versus another new image. And so the point here is that this is a recognition task. And so you don't have to recall anything about what recently happened. You simply have to be able to recognize what you saw and what you didn't. And so the way I always think about it is it's like when I see somebody and I'm like, oh man, that guy looks familiar, but I can't recall his name. And so I can recognize him, but I can't recall. And that's where the difference really lies. And so on this sort of task, it goes from your visual cortex and then just everything's done in the medial temporal lobe. It doesn't need further processing. And we see that accuracy on these sorts of tasks really are being reached, adult levels of performance are being reached at a very young age. And in fact, the activity of the medial temporal lobe goes down over time as it becomes easier. Okay, so after telling you all that, whoops. Okay, so something wrong with my slide here, but anyhow, in monkeys, it's a little bit different, but you see a similar thing on a spatial task like here. You can see, so in this particular study, what they did is they actually froze cryogenically depressed activity within the dorsolateral prefrontal cortex. And so when you do that in young monkeys, 9 to 16 months of age, it doesn't affect their performance on the task. And it's not until they're about 36 months of age that when you take the dorsolateral prefrontal cortex offline by freezing it, that performance is impaired, suggesting an increasing dependence of spatial working memory ability on the dorsolateral prefrontal cortex. And you see the same thing with activity. The number of neurons that fire during the delay period of a spatial working memory task doubles from 12 months of age to 36 months of age. And you don't see this on an object familiar task. In fact, here what you're looking at is accuracy in monkeys who are 3 years old, and it only required them a few sessions before they mastered the task. At 24 months of age, you see the same pattern. And even at 1 year, they're able to reach adult levels of performance with only one extra session of training. And so, the difference here is between the dorsolateral prefrontal cortex and the ventrolateral prefrontal cortex. The dorsolateral is maturing later in both monkeys and in humans. And so the limitations of working memory ability, especially within the dorsolateral prefrontal cortex during adolescence, is thought to reflect immature circuitry. And so if we take a slice here and we look at it over here, area 46 and 9 are considered dorsolateral, 45 and 12 are ventrolateral. And so if we take a look at the circuitry, take a traverse right through here, you can see the 5 different layers, and these are different GABA neurons. So a cholecystokinin basket cell, parvalbumin expressin basket cell, parvalbumin expressin chandelier neuron, all GABA neurons, interneurons, right? And so the triangles then are pyramidal neurons. And in layer 3, these are what are synchronized during the delay period and allow you to actually remember things. Information comes in through 3, and then this information is relayed to layer 5, and then the output is going to be whatever your response is. And so it's during adolescence in monkeys that you see refinements going on in this circuit. And so for one, there's an increased density of the axon varicosities of these parvalbumin expressing basket cells. You also see a decreased density of axon terminals from the fast spiking chandelier cells, a decreased density of the dendritic spines and excitatory inputs to these pyramidal neurons, decreasing 30%. And you also see increased levels of CB1 receptor mRNA, which is coming off these CCK expressing cells. And these increase by about 11% during adolescence in monkeys. Finally, you also see increased synthesis of 2-arachidonyl glycerol and DAG lipase alpha, which is necessary for the synthesis of 2-arachidonyl glycerol. And so these maturation patterns are strongly correlated with the oscillations that are generated by this circuit and required for the maintenance of working memory information. And so while these normally are going to strengthen the network connections and improve cognitive control functions, environmental factors like cannabis could potentially affect this and thereby impair working memory, as well as contribute to the occurrence of schizophrenia, which is characterized by very similar working memory deficits. And so our hypothesis was that there would be an alteration within these GABAergic neurons that help orchestrate this, the oscillations in this circuit. And we would see this selectively in the dorsolateral prefrontal cortex, but not in the ventrolateral. And by the same token, we expected to see that spatial working memory performance would be impaired, while we expected not to see that on a ventrolateral PFC dependent object working memory task. So I trained the monkeys and adapted, 2 working memory tasks that are commonly used from the, and this just shows you a monkey on a Wisconsin general test apparatus that Pat Goldman Rakesh had used. But I didn't have that, and so I used touch screens. And so we sat the monkey in front of a touch screen, and 4 locations would, one of 4 locations came up on the screen. And so when the monkey was there, one location was shown, he'd have to touch it. Then he would have to touch a center fixation spot so that he couldn't just hold his hand over the location, and he would have to remember it. And then if he did remember it and touch it, he received the reward, which was a drop of water. We also trained him on an object working memory task where one of 4 objects, 2 objects, 2 different colors, would appear in the center of the screen, and the monkey would have to touch that. And then after a delay, all 4 objects would appear, and the monkey would have to remember which one he saw most recently and touch it for a drop of water. And so you can see I trained 14 monkeys to do this. And then we divided them based on their performance in pairs to either receive THC or a vehicle. So this gets me to the next study. Here, looking at our 14 drug-naive rhesus monkeys at an average age of 28.6 months is when THC administration began. Again, counterbalanced based on their performance, like I just showed. And these monkeys, I surgically implanted a vascular access port as well that went into their jugular, and then we tunneled it to their back so we could access it daily. And we gave THC via the IV routes or vehicle, obviously. And then we tested a bunch of different doses. And what we found was that we saw both a delay and a dose-dependent effect. And so the delays are shown on the x-axis, and the decrease or the change in accuracy rate. So the animals were tested twice each day. We would put them in the chamber, have them complete the task, take them out, administer the THC or the vehicle. 30 minutes later, we brought them back to repeat. And so what you're seeing is a change between those 2 sessions within a day. And so we're just looking at the different doses of THC. The placebo monkeys were not included here, but we see a dose and delay dependent effect on the spatial delayed response task. We did not see that on the delayed master sample task. And in fact, we had 2 different delayed master sample tasks. They were identical except the colors of the objects were blue and yellow in one instance and red and green in another. And what that did was that gave us 2 levels. And so when you see the DMTS, that's just a delayed master sample that gave the same reward as the spatial delayed response task. Whereas the DMTS large, we gave twice the reward. And so we wanted to see if motivation was in fact impacted. And that's why we did this. But what you see is a very clear effect on the spatial working memory task across the different doses. And so it was based on this that we selected the doses we were going to administer chronically. And so what's important here is that these doses, obviously we showed acutely impaired spatial delayed response accuracy, but these are also doses that were shown to be self-administered by monkeys, known to produce effects in humans and comparable to smoking a joint or 2. And so here, what you see is the study 1 is that baseline study I told you about. And so you can see we had about 4 to 5 weeks that monkeys were required to get through that. Here you can see that our first pair of monkeys were a little less certain what we were doing. And so it took us 10 weeks to actually get through all those doses of THC. The next several pairs were much more uniform. And then by the 7th pair, I was quite tired of doing this and decided to make it a 5-week thing. Anyhow, after that is when the repeated dosing started. And so their performance now was, we only looked at their performance 23 or 72 hours after they were administered THC. And so it was always administered after we had them in the chamber. And so this is the period we're looking at next. And I'm going to speed up so I can get through this stuff. So delayed master sample, you can see from baseline to week 24, nothing happened, no difference between the 2 groups. On the delayed master sample large, the same thing, performance was better for both groups, but no difference. And what we saw here was this very clear effect on spatial delayed response. So at the 6-month point, we actually changed some things up on the animals. Replaced a 4-second delay with a 32-second delay. I removed the large DMTS trials. And on the spatial delayed response task, we kept the 4 choice, but we added an 8 choice. And so what this did in essence was give us 3 different types of spatial delayed response trials. One was the original or the O SDR, one where they had to respond to the same locations as the original, but included other distractor locations, I called them. And so D SDR 8. And then one where they had to respond to novel locations. And so N SDR 8. We also challenged them with some additional tasks, but I'm not going to go over that at all. And so the last study had stopped at this point. But these animals were administered THC for an entire year or 52 weeks. And so you can see that here. And so the next study of the original SDR 4-item trials spanned this entire period. And then for the distractor and novel SDR trials span this period, but I'm going to actually show you data that goes all the way out to withdrawal. The other points were where this paper reported to. And so on the original 4-item, looking at 16-second trials only, you can see that their performance actually improved. And these points here are actually because of all the changes that were made. It's a long story, but nothing there really. Their performance had recovered. On the distractor trials, there was a clear separation at the end of treatment, but by the end of withdrawal, it had gone away. And then on the novel trials, we actually saw that there was a persistent effect that went off during the withdrawal period. We also looked at different protein and mRNA levels, including the CB1 receptor. We looked at ABH6, which is a hydrolase that is co-localized with 2-arachidonoglycerol, and it metabolizes it. KCNS3, gene encoding a particular potassium channel that's necessary for the parvalbumin GABA neurons to function properly. And so I'm just going to skip over the methods and tell you what we found. We had reported similar findings in individuals with schizophrenia, but this data from the monkeys is actually not published. And so, because it is much later than I expected, I'm going to go through it quickly. What we saw was no change in protein, a change in CB1 mRNA in the dorsolateral PFC only. We looked at the laminar expression, and we found that it was only in the deep layers 3, 4 that there was a change in the expression of CB1 receptors. And this suggests that it was not affected on the GABA intraneurons, but in fact on the pyramidal neurons, which are also known to express CB1 receptors, though in much lower concentrations. Next, looking at the ABHD6, we didn't find any differences, and this data is in fact published in that paper, but this data is unpublished. Looking at the KCNS3, there was a significant difference of this particular potassium channel on these parvoalbumin basket cells. And so, putting all together, I'm going to skip through some of this and just get to the end. And so, we know that exogenous THC is going to affect all CB1 receptors, and it appears as though there was some change to the circuitry, and this may underlie the reason that repeated THC administration caused these persistent working memory deficits. And it also suggested that medications aimed at strengthening the network connections within the dorsolateral prefrontal cortex may help people recover. And so that's what brought me, in fact, here now. And so looked at guanfacine, which is an alpha-2 adrenergic receptor agonist. And the rationale was because of work showing that there's hyperactive neuroadrenergic system in rats following CB1 agonist administration. Clonidine reverses rimonabant precipitated withdrawal, and of course, Meg Haney's work showing lofexidine improves sleep and relapse. And there's a strong reason for looking at these particular types of medications for improving cognitive functioning, and guanfacine is better than clonidine and lofexidine for a few reasons, and we can't really look at that. But so we conducted a study, and I'm just going to have to skip to the end of it rather than tell you all that. So we saw that the THC by itself impaired performance on a dual MBAC task, and that guanfacine appeared to improve that. And the same thing on an attentional task. And so dual MBAC would be working memory, that the CPT task is an attentional task. It really only impacted omissions, but this was a very small sample size. We also looked at subjective effects. And so during the placebo, you could see that THC produced effects and the guanfacine had no impact, on any of these VAS ratings. We looked at cannabis withdrawal, used a few different withdrawal scales, and we really didn't see anything here either. There was a hint on a few things, but nothing really hot. And so very quickly, I'll just tell you about this study. We looked at CBD. These were domestic canines who presented at a local vet in Houston for lameness due to osteoarthritis, randomized to 1 of 4 treatment groups, CBD at 20 or 50 milligrams per day, a liposomal CBD at 20 milligrams per day, or placebo. A couple of assessments. The vet rated, and so what you see is that the 50 milligrams of CBD improved performance from day 0 to day 30, as did the liposomal 20 milligram on most of these. And this was similar to what the dog owners also rated the improvement. A few other studies, and then, so it's going to be a 2-site study. One is the VA, and the other is Texas Children's, looking at back pain and endometriosis, treat them for 8 weeks, probably just going to look at CBD, but maybe I'll tell you about that next time since we are late. The assessments, timeline, and thank you. Sorry.
Great, Chris, Dr. Verrico, thank you so much. That was Wonderful. Hearing about your trajectory to where you are now doing clinical trials with, it sounds like you'll be using CBD to understand if it might be helpful for chronic pain as well as pain associated with endometriosis in two very different populations as well.
Yeah.
Yeah.
It's, I mean, THC has a well-known analgesic mechanism. CBD doesn't, but it's a lot easier to study CBD than it is THC. So that's what we're going to do.
That's great. So I think we have time for one or two questions. So we have one from Dr. Chris Evans. And he was, it looks like he was reflecting on your experience at Sanofi with rimonabant. And he says, do you feel like there's hope for antagonist drugs other than inverse agonists, which we know rimonabant is suggested to be? Thinking neutral antagonists or NEMs. I think that's a NAM. Chris, I think that's supposed to be a NAM. Chris Evans.
Okay.
Negative allosteric modulator for the purpose that rimonabant was developed to treat.
Yeah. And so I definitely think that neutral antagonists, and it surprised me that Sanofi didn't pursue this further. And I do wonder if some of the minor cannabinoids act as, in fact, neutral antagonists, which would be great for obesity because you probably wouldn't anticipate some of the other effects. But it's a great question that I hope we know the answer to someday soon.
So, and because you've focused a lot in the preclinical space and animal studies, has there been much research looking at neutral antagonists compared to the inverse agonists in animal models? And have they shown any real differences between the two?
I recall one study that I saw recently, and I don't believe it was in vivo. I think it was ex vivo. And so there was they didn't look at any outcome measures that would give us that indication. They just were looking at mechanism. So it does appear as though they're available potentially, and they just need to be tested.
Thank you. And then we have one last question. Will monkeys self-administer THC?
Yes.
I guess there's the question about if there's a stressor effect of non-contingent THC administration that might have occurred when you were.
Well, that's exactly why, I surgically implanted the vascular access ports, and, the monkeys had no idea. I mean, they did, but, it was very painless for them. They didn't react at all. And, even if it did cause some stress, the vehicle animals were exposed to the exact same thing. And so, but yeah, no, I do not think it caused them any stress whatsoever. I mean, they were adolescent monkeys, and I don't know how many of you may have ever interacted with rhesus monkeys, but if you interact with the adults, they're very aggressive. But these little ones, man, they were so cute. They become your friends. You work with them 7 days a week, which you have to do that sort of study. And, you can't help it, and they know you and they groom you. And so, they weren't stressed by us at all. And they were, they're actually very healthy because we did interact with them so much compared to some of the other animals in the colony that, didn't get to interact with humans quite so much.
Right. But, thinking about THC and the non-contingent administration, the aversive qualities that it's reported to have in rodents and, the conditioned place aversion models. In humans, it's thought to, in some people, produce anxiety at high doses. So even without, having to handle them to administer the THC, I mean, is it possible that you were able to see some potential,
they were
stress?
High. You could see that they were high. I mean, in their eyes, it wasn't stress. In fact, it was the vehicle animals. And so I don't know if you remember, there was one spot on the one slide where I said, the spatial delayed response performance sort of fell apart at week 40 or something. And that's because when we changed everything up, we changed delays, we added tasks, we removed parts of some tasks. The vehicle animals were just like, what the are you doing? They were really stressed out about it, right? The THC animals were like, hey, this is great. We don't care. We're going to keep doing the same things we always do. And so, in fact, I would argue, no, it was the opposite, that the vehicle treated animals showed more stress than the THC exposed.
Great. Good. Well, thank you so much, Dr.
Thank
Verrico. It
you.
was wonderful to see you. Hopefully we'll see you in person sometime soon. But we really enjoyed hearing about your trajectory across species. I mean, not very many people have researched drugs and, also in canines to add to it.
Yeah,
So,
right. Yeah, no, it was great. I appreciate it. It was a great exercise for me. And thanks for listening.
Great. Thank you so much.
Bye.
Thank you all. We'll see you soon.